The Conversational Flow, a KCAS podcast.
Hello, and welcome to the third episode of The Conversational Flow, an official podcast of KCAS Bioanalytical and Biomarker services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcasts, the Weekly Bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam Cotty, and I've been in the flow cytometry space for over 15 years, and I'm here with my co-host, Brian Weil.
Thanks, Adam. Brian Weil here. So I've been in the flow cytometry space for over 12 years now, and extremely excited to keep working with my colleague Adam and all the talented personnel at KCAS Bio here in the Philadelphia location.
Thank you, Brian. Brian and I are members of a growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. While our KCAS headquarters are located in Kansas with additional locations in Europe, we are both here in Philadelphia, and Jeremy, our producer, is in Missouri, and so we're truly all over the map. We're excited to have you listening to the third episode of our podcast, which will be available virtually everywhere. Wherever you choose to find and play your podcast, you will likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will be a review of the latest news and resources, and then a focus on a topic of our choosing before discussing any feedback we receive from you. We're constantly looking for topics, and we'd be happy to discuss something that you want us to cover. So again, we're fired up to have you here, and we are looking forward to a fun episode today. To kick us off for episode 3, Brian is going to go over today's podcast topic a bit before we jump into the news and resources section. Brian?
Thanks, Adam. Our topic for today is, what is the KCAS Bio philosophy on flow cytometry validations? There are a number of different guidances around, and we'll go into great detail on how we perform validations, in what context, and why we do what we do. We've got two stories today that are around, in terms of medical research news, that are around advanced therapies. So one is the recent Moderna-Merck cancer vaccine readouts. And there we saw that when the Moderna cancer vaccine was paired with the Merck Keytruda monoclonal antibody, the rate of recurrence and death was reduced by 50%. So a pretty significant change versus the single treatment by itself.
And this is a personalized vaccine, right? And so what I was reading is essentially they look at the tumor that they excise and compare that to healthy cells, try to identify the mutations, customize the vaccine, and then that's what they're treating with, right?
That's exactly it. And that's why we need something like RNA that's fast enough to produce and is a robust enough manufacturing process that can be produced at a high quality to be able to do that per patient.
Yeah, and production right now, they're targeting at about 8 weeks for this, whereas they anticipate being able to shorten that up to 4 weeks.
Right. Amazing how fast they can get a customized medication out and produce according to FDA specifications.
Yeah, absolutely. Again, just highlighting, though, that there's a lot of historical science that's going into the generation of these vaccines, that there's a backbone that's already been tested. And so what we're doing is looking at the target itself to make sure that it is efficacious.
Exactly. Yeah, so definitely an exciting note there. Oncology remains one of the largest indications that we really like to treat in most of drug development. So ideally, this will add another tool to the toolbox of what we can think about when we target cancers. In terms of another sort of advanced therapy, the first CRISPR therapy was approved. So that's Casgevy from the Vertex group. So it did receive its FDA approval, which is exciting. and it really brings in a new paradigm in gene editing.
And this is for the treatment of sickle cell, right? And so looking at patients that are 12 years or older and essentially reducing the pain that they experience and utilizing a cellular therapy, so genome editing. And so to modify, essentially what happens is during the course of development to switch from a fetal hemoglobin to the adult version, let's say. And so what they would be doing with this therapy is enhancing the fetal hemoglobin to be able to balance out and provide that hemoglobin to the patient.
That's it. And they're doing that by using the CRISPR-Cas9 system, which, much like the RNA therapies we were just talking about, is a very flexible way to go in and modify the genome. So there you can target it to different gene sequences by using a piece of guide RNA. And that guide RNA has a specificity required to target the gene of interest.
And so highlighting with this, very similar to that Moderna vaccine, is that this is customized therapy. And so it's autologous, meaning utilizing the cells from the patient to be modified and then re-implanted for the purposes of enhancing the fetal hemoglobin.
That's correct. Yeah. So yet another one where it requires quite a bit of cell culture, some relatively advanced facilities to be able to do this in a sterile environment and then re-inject the cells. So definitely still some barriers to introducing, especially in Africa where sickle cell is most prevalent, but definitely a promising development for the future. Bluebird Bio had come out with their sickle cell treatment, I think 1 to 2 years ago. So this will only help drive prices down and introduce new options into the market. So always optimistic there.
Mm-hmm. Absolutely.
Jumping over to the investment side of medical news, the theme of the investments today is ADCs. So the first item is that the Pfizer Seagen transaction is finally closing.
And to clarify, ADCs, we're looking at antibody drug conjugates.
That's right, yes. So ADCs, antibody drug conjugates, are exactly what they sound like. Another molecule or functional effector that is conjugated or tagged to an antibody. That way you get the specificity of the antibody as well as the mechanism of action of whatever it's tagged to.
All right, kind of thinking of smart weapons, right? You're delivering it only to the place that you want it to go. It's a payload generally of some sort of a drug or something that would destroy the cancer cells. And so it homes to a place you get to pick what the target is. Generally, these are either tumor-specific antigens or tumor-associated antigens. Difference being that tumor-specific antigens are highly specific to the tumor, as you might imagine, so there's less off-target effect, whereas tumor-associated antigens could have some expression on healthy tissue, but it's at a much lower level, so you're still biasing the target towards the disease.
Exactly, and this really allows you to cut down on the amount of systemic delivery of some of these molecules, which is what leads to a lot of the negative impact of chemotherapy drugs in general. So by targeting the drugs just to the location that you want them to go to, you can significantly reduce the off-target impact there.
Mm-hmm. And so this transaction actually doubles the cancer therapy pipeline, I should say, of Pfizer by bringing Seagen on. So that, and I hadn't realized this at the time, that Seagen is a world leader in ADC technology specifically. And so, your reason for bringing that up at the outset.
Exactly right. So definitely Seagen is almost synonymous with ADCs. Really sort of pioneered the technique and are still the largest player in that market. So this is the largest transaction for ADCs. And it's an interesting trend. And my second news item is basically all the other ADC acquisitions. So basically, Immunogen has been acquired by AbbVie, Aurum has been acquired by BMS, Hansoh is partnering at least with GSK. So you see all these major pharma looking at ADCs and deciding to purchase companies, which is an interesting sort of difference as opposed to developing in-house. Part of it is just the business model for the companies that I've mentioned. A lot of them do quite a bit of acquisition, so that's not terribly unusual. But part of it, too, is the complexity of ADCs. Antibodies themselves are already, a relatively complicated thing to manufacture. If you then have to conjugate a payload to it, it drives that complexity upwards even more. So having this expertise is just as critical as having the molecular asset itself. So definitely see those acquisitions as being a primary way to bring that in.
Yeah, absolutely. And I know this is the investment section. We had already spoken some previously about medical research news, but I was interested in looking at the Moderna news, specifically from the eye of investments. I would say not giving any investment, advice here. And certainly had tried my hand at trying to pick stocks in the past and found out that I'm as bad as what you would read most people are at picking stocks. But I will say with that the Moderna news had led to not only a groundswell of increase in their share prices, but as well other companies that are working on mRNA technology, BioNTech being one of them, and there's a couple of others that were mentioned. So it's always interesting how that works. Positive isn't always just for the one company that shows the data. A lot of times the add-on effect of other companies getting that same boost.
That's exactly right. Definitely nothing here is in isolation. So as soon as one ADC company gets bought, then you start to see a groundswell of other companies getting acquired, which raises their prices. Or as we said, in the Merck case and Moderna, basically they've proved out that is an efficacious therapeutic model. So then that makes everyone else working on similar models ideally go up as well, assuming that they've got a roughly similar process there.
Right, and you're seeing the same thing right now with cell therapy companies. There are a large number of them out there, and anytime you see news, you see presentations of data, it has this similar effect as we were just talking with about the Moderna effect there.
Right. And, those are the two primary medical research news and investments. So we'll run over into KCAS news. So here, it's the holiday season. So, of course, the biggest news really has to be our holiday parties and some of the community outreach that we've done. So we were able to sponsor a family locally, and that's what we've done at all of our sites here at KCAS to make sure they have an enjoyable Christmas holiday. And then we've had a couple gatherings for our staff to have everybody have the chance to get together outside of a work setting and, really just give thanks for the whole year. We have had a phenomenal year. So definitely appreciative of everyone's hard work toward that effort and looking forward to whatever next year brings.
Yeah, talking about the year, certainly a lot of work got done. Supported many projects, all sorts of indications, as well as we've talked about in our prior episodes. Bringing on new instrumentation and changing things up a little bit to support. The increase in growth that we expect and are looking forward to in the coming years. But I couldn't do it without the team. Really work with a great group of people here. So it's very exciting.
Indeed. All right. And now I guess we'll move over to our paper of the month. So Adam, you actually selected this one. So the title of it is Validation of High Sensitivity Flow Cytometry for Reliable immune cell analysis in real-world laboratory settings.
Yeah, I felt like it dovetailed really nicely into what we wanted to talk about today. We've alluded to this in the previous episode, specifically about validations. And, this talks specifically a high specificity or high sensitivity, flow cytometry, something that certainly by the nature of looking at populations that are below 0.1%, we've delved into at times, but there's challenges there, right? And so it was good to see a lab describe how they've gone about it, utilizing some of the same tools that we do, specifically the CLSI H62 guidelines. And really trying to put together appropriate test scripts to make sure that the assay can be relied upon appropriately, right? And so I think we'll plan on going into what kind of test scripts they talked about, what we generally do, as well as what can be added there.
Okay. And, I like this paper a lot because they were, of course, looking in a real world laboratory setting. So a lot of our flow cytometry panels get included in clinical or preclinical trials. So always a concern on, yes, it performs well when it is, drawn and then processed within 2 hours, but will it perform the same at 24, 48, or 72 hours, whatever might be needed by logistics? And I was very interested in how they were setting themselves up to answer that question as well.
Yeah, absolutely. And so I think the first thing that they mentioned right at the outset is doing the lower limit of quantification. This has been a test script that more and more we've seen over the past several years. When I started in flow cytometry CRO, specifically with Flowmetric, it was not something that was typically requested. More so because at that time really weren't looking as much at the rarer populations. Most of the panels were immunophenotyping, and I don't want to call them basic because certainly we had a compendium of markers that we're looking at, and each one was unique. As we've talked about, we do fit for purpose assays, and so certainly defined in that way. But with that, now we're seeing more and more that we're looking at very rare subsets, and so wanting to figure out how low can you reliably go here. And a lot of times too, there are things that you would do to ensure that you can get the numbers you want, and part of that is making sure the input, the amount of sample, the number of events that you're collecting is sufficient. And so many times that's one of the primary shifts for an assay that's going down this low, is to make sure the input number is sufficient.
And I like it too because, this is a very common problem. Yes, they're applying it to PNH, so paroxysmal nocturnal hemoglobinuria or whatnot.
Mm-hmm.
So PNH, that's why I don't pronounce those words. But this same sort of philosophy can be applied to a number of different settings. So definitely applied to minimum residual disease MRD assays, or also some of the PK testing for cell therapies where you might have really low numbers of some types of cells. So definitely see the same techniques and the same philosophy needing to be applied in multiple contexts.
Right. Certainly agree there. Very broadly applicable there. And so I think that certainly brings us to, what are the general categories of validation?
Yeah, absolutely. So here at KCAS Bio, we've separated validation into a couple different levels. If the top one is, of course, a full validation. Then we've got a couple different levels of qualification. And these would be reducing the amount of QA oversight or the amount of test scripts that we include. And then at the very lowest level, we would do sort of an exploratory optimization, potentially with a verification, as opposed to even a full qualification.
And again, this is evolving, right, or has evolved from where we were years ago, where it was a validation or really it was not. You optimized or you validated, but that doesn't suit every client and it doesn't suit every study. Generally the guidance is that if you're doing phase one study or exploratory endpoints in that typically go with the qualification. If you're looking at something that you want to expand that panel for use for later on, later phase work, then certainly you want to validate. Any other thoughts on there? Anything I'm missing?
No, I think that's exactly right. Basically, if you're planning to submit the panel to the FDA or results from the panel to the FDA as a primary endpoint or anything where they really need to see the rigor of the assay, we do still recommend a full validation.
Mm-hmm.
But as you said, for phase one exploratory endpoints, frequently qualification will be sufficient, and in some cases, a very brief verification would also fit the bill there.
And then generally too, with qualification versus validation, there will be more test scripts for a validation. Obviously, you've got your core test scripts that we see almost every time. Intra-assay, looking at the same sample closeness of results over and over again, just making sure that they're right in line with one another. Inter-assay, looking at changing laboratory conditions there. And so those generally make up the core of how is the assay running from a day to day and run to run. Then from that, we add in a number of different options depending on client request. Certainly inter-analyst, so you want to be able to have a number of scientists be able to run the same assay and be comfortable, because if you're tied into just one person running it, logistically, it's very difficult. And also talks to how reproducible that assay is in and of itself. Stability is another big one. So you have process stability. And so looking at the samples once they've been processed, how long until they have to be run. There's pre-process stability. So essentially for fresh samples, looking at when they've been drawn until they arrive, how long is that duration suitable before you can't count on the data, or certain populations might not be as reliable anymore. And then long-term stability too. How long, if these are PBMCs you're storing, or if they're fixed samples, how long can they go over time? What intervals are you looking at? And so those are the few of the my favorite ones. Really the core of what we do, and then we touched on several for the highly sensitive assay. What about you, Brian? What are your thoughts as far as test scripts there?
Yeah, I mean, you're exactly right that our full validations do include inter-assay, intra-assay, inter-analyst, inter-instrument, and process stability, as well as sample stability. And if it is a PBMC assay, it will usually include long-term stability as well.
Right.
So we do regard those as sort of the core parameters that need to be included in a full validation.
Mm-hmm.
And then really anything else that we layer on is going to be sort of context dependent. So if we are looking at a rare cell subset, then definitely limit of quantification or limit of detection will be critical ones to assess. And we can make other recommendations based on what the intended use is, in case we need to evaluate interference or anything else that may be going on in the patient that could have an impact on the assay itself.
hmm And you brought up the inner instrument. That was one I'm realizing I did not bring up. So looking at how close different machines are, and that's, always something that you want to consider. Machines can be set up to be essentially identical, but then have some variance there. You want to understand what that variance is. I think that is something that we're working to address more with spectral now, and those machines being specifically set up and harmonized more so than with conventional flow. But I don't anticipate that we would see that inter-instrument test script go away, certainly. It's always good to have eyes on that data and have that flexibility. So, from, again, talking from an operations perspective, if you are tied to one machine, but there are several assays that are tied to the one machine, it becomes a logjam, and you could eventually run into a stability issue. There's not doesn't tend to happen, but certainly looking out for any contingency there.
Sure, there's definitely instrument congestion, but then also instrument downtime, right? Pretty much all instruments at some point need to have preventative maintenance of some sort, or may have some type of issue that crops up and takes the instrument down for a day or two. And in those cases, you want to have a backup that will not alter the results of your trial. So definitely inter-instrument has that benefit as well.
I was wondering, so I hadn't really heard it termed instrument congestion before, and I'm wondering if that has to do with your current status, or
That's it. Still recovering a little bit from a little cold here, and bleeds into other areas of the lab, I guess.
For sure. Instrument congestion. I like it. I mean, it certainly makes sense. So, be clear, not being critical, but interesting way of putting it.
Yeah. As long as the Fortesses don't start sneezing on us, it's fine. No worries.
No. They don't want that at all, right? Have to call the technician doctor.
Indeed. Yeah, and then just to keep jumping into some more test scripts, one of the other things that I noticed from the paper that we had initially talked about is that they made their own control material.
Mm-hmm.
And I found that to be an interesting aspect, and something that we do here sometimes as well, by either identifying a control material, purchasing a very special control material, custom manufacturing a control material, or in some cases also then taking a whole cell fraction and enriching it.
Mm-hmm.
And definitely, I'd say that type of test script, or also using stimulated control material, can be critical to making sure that we're looking at the right thing for the assay validation.
You mentioned something there, and that's always a talking point in stimulated. I did want to address first, you mentioned about test scripts that they had looked at. One of the ones that we hadn't touched on but was in the paper was looking at carryover, from sample to sample carryover. Generally for things that are more highly represented, higher percentage populations, carryover is much less of a concern. Generally there's an accepted range of carryover. Between wells, there's washing that goes on to minimize that. But when you're looking at something that is under 0.1%, for example, in this paper, you do want to understand the carryover that you're getting, and so make sure to factor that in as you do analysis. With that, you mentioned about stimulation, and the sample type for validation, I think, is always one of the biggest questions that we get at the outset. Ideally, you would love to be able to do a validation on disease state, specifically patient samples, but that's not necessarily the case. Obviously, there's approvals that need to happen on patient samples, and so it's not something that you would generally get. More often than not, you could either purchase disease state samples. If they're frozen and you're going into a frozen matrix or a fixed matrix, that's easier. Whereas if you're talking about something that's fresh, a lot of times the lead time for that is fairly significant. And so a way around that is to go towards stimulation. With that, though, certainly stimulation presents its own challenges. And so, I want to throw it over to you, Brian, there, as I know we've discussed many times. What are your thoughts on stimulation?
Definitely a challenging part that needs to be addressed during the development, right? Where you need to know which type of cells you're stimulating, how much you're stimulating them, how long you're stimulating them for, and make sure that that's reproducible. So, we've done things like PMA and ionomycin, which are sort of broad stimulation conditions, or also things like C3, C28 that are sort of more focused on one area of activation or another. Mm-hmm. But definitely the type of stimulation that you're looking at needs to be targeted to the cell populations that you're trying to activate, because each one will have a slightly different reaction to different types of stimulation.
Yeah, absolutely. I mean, and there are all sorts out there in addition to those. You got pokeweed mitogen is one that sticks at the top of my head. I mean, can't forget that. They're trying to remember several others as well, but was it the more the peptide based, and so specifically looking at bacterial proteins. Why am I forgetting the specific ones? Anyway, I'm sure that'll come to me right at the end of the podcast, as always. But, it's that whole being put on the spot thing. And of course, it wasn't you. It was myself putting myself on the spot and then blanking. So apologies to the audience there on that one. But there are a wide variety of stimulants that can be used. And it depends on what the target cell type that you're looking at, because they don't all stimulate the same. And also the duration, as you pointed out, is key, because you go too long, you get high cell death, introduce additional variability. And the other thing that is key is, you have donor variability as well in response to these stimulants. And so that's where another thing that you mentioned has to be taken a look at in the optimization phase, so that you're setting yourself up for success to really assay. You're looking at the assay performance, and you're not necessarily looking at the sample itself as far as how it reacted and caused problems. Now, of course, it all brings up the point that some disease states are very heterogeneous, right? You're thinking about AML, for example, where patient to patient, time point to time point, you can see changes. And so it is something that going through the validation, you try to fit it for purpose as much as you can. But there's only so many samples that you're going to run in a validation, various considerations being cost, time, so on. And so, kind of babbling on there. Any other thoughts, Brian?
Not with regards to stimulation and other test scripts in general. I think we already covered some of the primary ones in terms of LOQ, LOD, LOB, and long-term stability time points depending on how the trial logistics are set up. So I think that is a pretty good coverage. I guess the last aspect is, now that we've got both Cytex and Fortessas, are there any differences between spectral and conventional flow validation?
As far as the test scripts are concerned, it's essentially the same, right? I mean, you got to make sure that you get your controls, you get your spectral and mixing considerations there. But when you're talking about the test scripts, you want to again understand how the assay is performing. So you're going to do your intra-assay, your inter-assay. You want to understand the difference between analysts, if there is a difference, whether or not the instruments are different themselves. and then stability of the sample. So those core really are going to be very much the same as to the offerings that you would have on a conventional flow cytometer.
And take roughly the same time.
Mm-hmm.
Yeah, I mean, I would agree with that overall. I would say that, as you mentioned, the unmixing panel does need a little bit of optimization.
Mm-hmm.
So looking at if you need a bead or a cell control.
Right.
So generally we assume 1 to 2 additional days on the development time frame.
Mm-hmm.
But otherwise, very, very similar in terms of total time for validating.
Mm-hmm. And it was SEB. That was another one of the
There we go. I knew it would come back to you.
Knew one of them. There's still more bouncing around in my brain. So, when you get those kind of things where it's a little bit hazy until all of a sudden it comes in front of your brain, and you're like, oh, wow, there it is.
Maybe there'll be a new running gag. Every once in a while, you can just shout out a random stimulant condition.
Either that or I need to get a fidget toy to keep myself on track. To your point, absolutely. There are more complexities with spectral flow, and we need to keep those in mind. But the understanding of the assay itself, the test scripts, really shouldn't change very much. Don't change very much from a spectral to conventional.
Okay. Excellent. Anything else you wanted to add on validations overall?
Really that it's fit for purpose. I think the discussion needs to be had up front. What are these going to be used for? I know it kind of pointed out that at the beginning, but it bears repeating that you really need to understand what it is that you're looking at down the line to know which reportables are the ones that you're going to want to have validated. Is it acceptable to have other ones that are more exploratory reportables, specifically because maybe they are very low frequency and you don't think that there is going to be a likelihood of it validating or something like that. But when you craft it, build the validation plan, then everyone's on the same page as to the expectations. It goes nice and smooth that way. When there's missing information as to how it might work on the back end, that's where sometimes it doesn't go as smoothly. And so that's certainly our team is trained to ask these questions, right, all the way from the business stage on through to the lab to make sure that clients are getting the validation or qualification that meets their needs.
And that's exactly it. We understand sponsors want to basically run as many samples as they can because that's, what they are really there to do. So we had to balance that requirement to, spend more on sample analysis than validation against the requirement to make sure that we're not just generating crap numbers and that we can, give them data that they can be confident in and can drive decisions.
Mm-hmm. Absolutely. Yeah, so that was the question for me. I mean, I think we covered what we wanted to talk about. Any other thoughts on your end?
No, I think that does it.
Sounds good. So, we're coming up on the holiday, and we got what is it? Christmas on Monday, right? And so, anything specific that you're looking to for the holidays, Brian?
So we're going to do a big neighborhood gathering with like a hot chocolate bar. So I've got like a slow cooker hot chocolate recipe that is pretty awesome. Eggnog all around. So have a gathering with all the neighbors. And then some families actually in from Alaska. So we're going to hang out with them during the Christmas season here.
Oh, wow. From Alaska. So what brought them to Alaska?
Yeah, a winding road.
That's great.
Yeah, they actually worked at a ski resort up there, and it's one of the bigger ski resorts in Alaska. So if you live in Alaska, there's a decent chance that that's what you do. If not oil and gas, then probably this. So yeah, and they thought they were only going to be up there for like a summer, and they decided to just stay. So now I think they've been up there for 8 years, 10 years, a long time. So.
Yeah, it's just because Alaska is definitely beautiful. My wife and I went there on our honeymoon and really appreciated it. It was amazing that in the the beginning of June, I think the high got to 70 one day, but it was pretty much averaging in the 50s. And so that's a far cry from when you're used to it being 80s, 90s every day. But yeah, certainly very beautiful. And I think the thing that stuck with me was we were in Juneau and we did a nature hike, and the guide was giving us various facts. And I think one of the things that he said was about bears. And I may not get the exact number correct, but he was telling us like for every one bear that you see, there's at least 5-10 bears that have seen you and you don't even know about it. So that had me looking over my shoulder a little bit on that hike.
I believe it. Alaska is definitely lousy with bears.
Yeah, seriously. So as far as on my end, typically travel to see family. So looking forward to doing that again. But it's been interesting with, kids getting older and shopping and whatnot. There's just less hiding of anything, and in fact it's more of a collaboration on gifts than anything else, which it's kind of fun. I still enjoy it. It's just a different experience. When my daughter was little, she was intent on proving that Santa wasn't real, and so she put her phone with video record pointing at the tree on Christmas Eve, and I was downstairs, I was wrapping the gifts, and I see the phone right as I'm coming up the stairs, and so without getting into the frame, I pause it, put all the gifts down, and then unpause it so she never saw me. Got all excited to see it, and then she goes and looks at the video the next morning. She's like, "You paused it. I know you had to have done that." But we laugh about that every year.
Good times. Very sneaky as well to have put the phone down there too.
Yeah, I was glad I caught it because it wasn't immediately obvious. But always got to keep on your toes with the kids,
And I guess with that, we'll go ahead and close out this year's podcasts. Our next podcast we'll be looking into considerations for spectral flow as part of that podcast.
Yeah, looking forward to it. Definitely a fun topic.
All right. Thanks all, and we'll talk to you next time.
Take care, everyone.